Question number 7. What are the functions of the following organelles? So the endoplasmic reticulum, we'll start with that one. So the endoplasmic reticulum is the site of membrane protein synthesis. So all membrane proteins in our cell get inserted into the membrane at the endoplasmic reticulum. All right. In addition, at the endoplasmic reticulum, some proteins get glycosylated. So this is a new term for you guys. So glycosylation means putting sugars onto a protein. All right, so that chemical reaction. So certain proteins in our cells need these sugars in order to function. All right, and so those sugars get appended, at least the first round of them get appended in the endoplasmic reticulum. All right, the nucleus. This one's pretty obvious to you guys. The nucleus is the site of DNA storage inside of our cell. So if this is where the DNA is, then it also has to be where RNA gets made. So all RNAs are made inside of the nucleus as well. Mitochondria. This is the main energy production center of a eukaryotic cell. For now, that's all you need to know about the mitochondria. Later on, you will know the biochemistry of the mitochondria inside and out. All right, D, the Golgi apparatus. So the Golgi apparatus kind of comes after the endoplasmic reticulum in terms of processing proteins and other molecules that are headed towards the outside of the cell. All right, so the Golgi has basically two functions. Right, first of all, it can add more glycosylation onto proteins. Secondly, it packages membrane proteins and other molecules into vesicles that can then be trafficked to other areas of the cell. All right, so in general, you can kind of think of this like a membrane protein, for instance. It gets initially made in the endoplasmic reticulum, then gets transferred to the Golgi apparatus, and from there moves to the cell membrane through what's called a secretory vesicle in this case. All right, vesicles, all they are basically a sphere inside the cell that's surrounded by a membrane. So basically a Golgi apparatus kind of buds off these secretory vesicles that can then go and fuse with the cell membrane. All right, now the lysosome. All right, the lysosome is a type of vesicle, but it's one in which large particles from outside of the cell get brought in and kind of blebbed off into this vesicle inside of the cell. So this is our cell membrane here. We have some big protein, all right, that binds to it. Well, initially, this protein is going to kind of push down here or bind to receptors, and there are a whole bunch of chemical processes involved with this. But essentially, now the membrane is going to kind of bend inward, and eventually, the invagination is going to break off into a vesicle. All right, now this vesicle can move inside of the cell, all right, and it becomes a lysosome, all right, when this certain proteins on this vesicle start pumping in acid in here. All right, so the pH inside of this vesicle decreases dramatically, and it becomes what's called a lysosome. So the function of a lysosome is to digest proteins and other large molecules that are coming in from the outside of the cell. So lysosomes are involved in getting our cells food from the outside environment. All right. Then lastly, we have chloroplasts. So chloroplasts are in some ways similar to the mitochondria. There's a lot of belief that both mitochondria and chloroplasts derived at some point from a prokaryotic organism that was actually eaten by a eukaryote. There's a lot of reason to believe this. For instance, both mitochondria and chloroplasts have their own DNA. They both also have their own ribosomes, stuff like that. All right. Chloroplasts, though, are different from mitochondria in that they use the sun to basically produce energy. All right, and they can use that energy then to make ATP, or they can use it to store that energy as sugar.